So now that we've seen the anatomy of these chromosomes, what we're going to do is to ask the question, what do they do? What's the physiology of the chromosomes? It's a very complicated question that we're going to be spending most of this class actually answering. And I want to start by looking at what the chromosomes are going to do during development, because what's happening really is that the chromosomes hold on them the program, like a computer program, if you will, of how to build an organism. The basic procedure, the basic process that they cause is very similar among all animals. In fact, actually remarkably similar among all organisms on the planet, which means that many organisms have the same genes. This becomes an interesting sort of pattern that we're going to study later on. But to start with, I want to look at the development of an embryo, embryonic development of a mammal. And this is basically true of essentially all the vertebrates, all the organisms with a backbone. These organisms, mammals included, start out as what's called a zygote, a single cell, one single cell. And it has a complete set of chromosomes. A human zygote, therefore, would have 46 chromosomes, actually 2 sets of chromosomes, so 23 pairs. And it starts when a sperm cell, which is carrying 23 chromosomes, fertilizes an egg cell, which is also carrying 23 chromosomes. And those chromosomes then fuse at the fertilization event when the nuclei of those two cells, the sperm and the egg, fuse. And those 23 chromosomes constitute then a diploid zygote. Remember, diploid means 2 sets of chromosomes. Now, what happens after that is a series of divisions that we call cleavage divisions, because imagine this zygote, which is a relatively large cell, just gets cut in half by a cleaver. And it gets cut in half, and it doesn't grow before it gets cut in half again. What happens is the zygote gets cut, the cells stay the same size, and then it gets cut again as quickly as it can. But in this case, it gets cut at exactly right angles. So the first cut is like this, and then the second cut is at right angles to that. And then it gets cut again. A third set of cuts is right at right angles to the first two, because remember, we're in three-dimensional space. And each time these divisions occur in the cells don't grow in between. That's what defines the cleavage divisions. We're just cleaving the egg down into pieces. After a certain number of those divisions, just a few, the cells then between divisions will start to grow. They'll grow back to their original size. Because remember, you start with a cell like this, it cuts in half. When it divides, it has to grow back to its original size. When that starts to happen, we then transition into the next stage of development. We get out of the zygote stage and we go into what's called the morula. And the morula is just a solid ball of cells where the cells are dividing. It's getting bigger and bigger and bigger. And then something interesting happens. The morula, which has a bunch of cells in it that are what we call totipotent. These cells, if we were to pull one of them out, we could actually make a new organism that's identical genetically to the original organism. This is a way of making twins, for example. That morula will start to fill with water. It's a solid ball of cells, and it'll start to fill with fluid and fill with fluid and fill with fluid. It does that because the interior cells start to secrete salt, chloride in particular, and the water moves in by osmosis and pushes the cells out, and you end up then with a hollow sphere like this. Once you get to the hollow sphere stage, you're in what's called a blastula. Now, none of these are embryos. The zygote, the morula, and the blastula are not embryos. They're something different. The blastula is still filled primarily with totipotent cells, cells that if you were to take them out, you could grow them into any other tissue and therefore into an entire human being. In fact, that's exactly how identical twins arise. Either the morula or the blastula splits, and those two things then grow. They have identical chromosomes, and therefore they grow into identical twins, which are clones. Now, when the blastula continues to develop, you'll see something else very interesting begin to happen. And at that point, you can no longer have identical twins. Because what happens is the cells will start their programs to specialize. They'll start to become specific things. And you'll see the very, very first structure form, which is usually a puckering on one side of the ball of cells. And that puckering is going to form the gut. It's going to form the intestines and the stomach and so forth of the organism. But also other cells are starting to differentiate, starting to change their structure, and they're starting to commit themselves to becoming specific types of cells. When you take developmental biology, you'll see that this beginning to change will start making 3 different layers: the endoderm, the mesoderm, and the ectoderm. And each one of the cells in those new layers are now committed to becoming specific types of things. For example, the endoderm cells, which are shown in yellow here, are going to become the gut and some of the accessory structures to the gut that are going to secrete things into the gut during digestion. The mesoderm will give rise to things like muscles and tendons and so forth. The ectoderm will give rise to the skin and the brain and things like this. So once the cells get to that point and they start committing to specializing, they no longer can form twins, because if I were to take an ectodermal cell out, I couldn't then make it become, without working very hard, I couldn't make it become a cell in the gut, for example. And that means then that once we get to this stage, we're committed. This is not going to be able to form a new clone. It's got to completely develop or it's not going to make it. Once we get to that stage, we call that a gastrula. Okay, so first we start with the zygote, then we go to morula, blastula, gastrula. The gastrula continues to form, and then when you start to see structures, for example, you start to see the limb buds start to form and the eye begin to form. And you can start to identify the structures, but a non-expert cannot tell what species it's going to become. Then we're into the embryonic stage. Right? So the embryos then will look something like this. This is a picture of embryos that's written, that was actually made by about the turn of the 20th century by a German, or actually a Prussian biologist by the name of Ernst Haeckel. And he was very interested in looking at these embryos because he noticed that if you look at all these different embryos, they have a lot of similar structures. In fact, they're so similar that, again, a non-expert can't really tell them apart. And here's an example of that. We get to this point here. If we look, you can see every single one has an eye structure here, and then they all have tails. And if you look, they all have these things around in their, what is going to become their throat. And they always have the same number of these little folds right here in those tissues. Now, what are these things? Well, if you go a little bit further in development on this lineage right here, this thing becomes this, you'll see this is starting to become something that looks like a fish or something like this, or a salamander. But anyway, you can tell those structures, those folds in that throat area become gills. And that's true then when you actually get to the fish. This is an amphibian, those are gills. And so again, these are gill arches. That's what we call them today, because they are the same structures that arise in fish as gills, and they're also constructed by the exact same genes that makes gills in fish. So they're gills, they're gill arches. But these creatures, well, they're not fish. So what are they? Well, if you look at them again, you can't really tell them apart unless you're an expert. An expert would be able to say, okay, well, this is that, and blah, blah, blah. But this set of embryos right here, even though they have gill arches, are not going to have gills as adults. That was another thing that interested Haeckel quite a bit. If we continue with development, we go out of this stage where you see structures like limb buds like this and so forth and tails, and you start to be able to differentiate what they are in your own mind. You can tell, okay, this is going to become this organism, this is going to become this organism. When you get to that point, then we transition from embryo to fetus. A fetus is a developing organism, the species of which a non-specialist can differentiate, can tell. And here we are. Here are the fetal stages of all of these different organisms. See, this became a fish, this became a salamander, but this lineage gave rise to a tortoise. This lineage gave rise to a chicken, this to a pig, this one to a cow, this one to a rabbit, and this one to a human. Now, if you look at these, look at this. The bird, the pig, the cow, the rabbit, the human, they all look very similar, and they all have gill arches. Well, wait a minute. Humans and pigs and rabbits don't have gills. Well, actually, we still kind of do in a sense, but they don't function as gills anymore. This is the top one. The first one is this one. It's the maxilla. And then this is the second gill arch, the mandible. And then you've got gill arches that form little tiny bones in your ear, and the rest get totally reabsorbed. They get totally destroyed during the developmental process. But here you are, as a beginning human, they have gill arches. Also, notice you have a tail. Right? See, there's the tail. Do humans have tails? Please say no. We don't. Occasionally, though, a person will be born with a tail. You can be surgically removed at birth, no problem at all. It's a very simple procedure. But occasionally they have tails because the embryo has tails. But look at all these embryos have similar structures. That's a fascinating pattern that we're going to have to study later in the class. Why is it that we have gills at the beginning? Why is it that we have a tail? We have all kinds of other weird structures like that too, which we have to try to explain and understand. But the point is this: during development, we start out just like essentially all other vertebrates as embryos. And that's why the embryo stage is the one that's defined as not being able to tell exactly what they are without being an expert. But by the time you get to a point where you can tell without being an expert, then we're into the fetal stage. Okay, so to remind ourselves what's going on, we start out as zygotes, a single cell, go through a series of cleavage divisions. When the cells start to grow in between those divisions, we become a morula when it's a solid ball. When it becomes a hollow ball filled with fluid, we go to the blastula stage. When the cells start to commit to a specific type of lineage and we start to see the first structure, we go to the gastrula stage. Then we go to the embryo stage, and then finally this fetal stage. Okay, so all of that development is very natural, and it happens without any sort of outside thing pushing it. Everything that is required to occur is coded for in the DNA. But let's think about exactly what has to happen here. First thing, you've got to solve the problem of the number of cells. When you start out, you started as a single zygote. There it is, one cell. But then you have to give rise to a bunch of cells. How many cells do you have to give rise to? Well, a person my size has about 30 trillion cells in their body. So one cell has to give rise to 30 trillion. How does that work? What do you have to do that? And what's interesting about it too, there's a second part of this problem. That is this. If you look at every single cell in my body except for a very few, and you compare them genetically, they are not 99% identical. They're essentially 100% identical. They're completely and utterly the same basic set of DNA. So that means this. Every single cell in my body, except for a few, the ones that are going to give rise to sperm cells, those cells, all of them, are houses of a complete set of human genes. Actually, two complete sets of human genes. So when we make these cells, we not only have to make the cells, we have to put a complete set of human genes into each of the daughters. So that's the idea. How do you do this? Where does that arise from? Well, that process is referred to not as mitosis, although you're going to see that a lot in textbooks. Nowadays, we call that proliferation. Cells proliferate, they divide. It's not cell growth. Cell growth means a cell gets bigger. Proliferation means that they divide. So you have one cell divides into two, just like we saw, and that happens over and over and over again. How many times does it have to happen in order to get the 30 trillion cells? Actually, only about 40, 45. That's all it needs. And you might think that's impossible. How can you only have 40 cell divisions give rise to trillions of cells? That's the power of exponential growth. We'll talk about that later. But the fact of the matter is that the cells are simply dividing, but every time they divide, they have to take all those chromosomes, all that DNA and genetic material, copy it, and put a copy into each of the two baby daughter cells. That process is called mitosis. So mitosis is not cell division. Mitosis is division of the nucleus. It's dividing the nuclear material. It's dividing all those genes and separating them out so that they can go into the opposite cells. The cell process by which the cell becomes two cells, actual cell division itself, is called cytokinesis. Again, this word C-Y-T-O, cyto means cell. Kinesis, kine means activity. So in essence, this is just saying the cell is actively dividing at that point. So if we look then at the cell proliferation cycle, there's two different processes going on here. There's the mitotic process by which the nucleus divides, and the cytokinesis process by which the cell contents themselves divide. Okay, well let's take a look. Let's just take a look at a cell that's in your body right now. Okay, just any random cell. It could be in your liver, your brain, wherever. First question, how many times is it divided? Less than 40 typically. Okay, now the question is, what's it doing right now? Is it going through mitosis, or is it going through cytokinesis? Or is it going through or doing something else? The answer to that is almost 100%. Unless you're taking it out of the bone marrow or the lining of the stomach or something like that, that cell is not dividing, so therefore it's not in mitosis, it's not in cytokinesis, it's doing something completely different. In fact, the vast majority of your cells in your body have essentially not divided since you stopped growing. They're basically just sitting there, not doing anything. And so mitosis is something we teach, but it's unfortunate because when we teach it and we emphasize it so much in classes, we make it seem like cells are constantly going through mitosis, and they're not. Even tissues that are growing extremely rapidly, like for example cancer tissues, the number of cells that are actually in mitosis at any given time is a very small fraction of the total number of cells that are in the tumor or in the tissue that's growing. So What are cells doing when they're not dividing, when they're not going through mitosis or cytokinesis? Well, they're not just sitting there really, they're being cells. So let's take a look at that. I want to look at the life cycle of a cell. I want to look at this like, for example, the life cycle of a human being. Human being, you're born, so you're a newborn, and then you're basically an infant, then you become a toddler, and then you become a child, and then an adolescent, and then a young adult, middle-aged adult, old adult. That's the life cycle. Now, if we look at the life cycle of a cell, it's different. It has similar sorts of properties. The cells mature and they change over time, but what's interesting is that the cell life cycle is quite a bit different than the human life cycle in a very fundamental way. I'll explain that in a moment. But in this chart right here, this chart is sort of showing what the cell is doing in its life cycle. And right there, at the end of that blue arrow, is where a cell is born, a baby brand new daughter cell. And it's sitting there and it starts to mature and it starts to grow. Now, that process of first growth and responding to its immediate birth so that it gets back up to its adult size is what we call G1. It's the first gap phase. You can think of the G, though, as growth. It's growing. And what it's doing is basically preparing itself for its adult life. So, again, it's sort of like a human being. There's the infant, there's the child, there's the adolescent, and so forth. It goes all the way up. And then it does something really, really interesting. Once it gets to be its adult size and its adult sort of life begins, it does something that you cannot. It stops aging. It really, literally stops developing. It stops maturing. We can't do that. Human beings can't do this. So this is very counterintuitive to the way we think. The vast majority of cells in your body do this. In fact, they're programmed to do this. They're programmed to stop aging. When they do, we say that they've gone into a process that we call G0 or G0. So they go into the first gap phase, G1, and then they're programmed to go into G0. And I see this arrow here sort of is represented as kind of this light dotted outline. That is absolutely wrong. The cells go into that unless they're told not to, or unless they're pathological. So the cells go in there and they'll sit like that. How long can they sit like that? They can sit like that for 80, 90, 100 years. They can literally not do anything further than just be a cell, a mature cell for that long. Now, you might be saying, wait a minute, that doesn't make sense. They stop aging. Well, they do. Now, that doesn't mean they don't take damage, and they do over time take damage. So your body will age, but the cell itself is not aging. It's not maturing any further. But you, as a result of all these changes that are happening in these mature cells, can age. So that's something we're still studying. But the point is, cells are very, very different. They can stop aging. And they'll stay like that unless they're told to start to divide. Now, here's an example of how that might happen. Suppose you're walking along and heaven forbid you cut your skin for some reason. Now, the cells in the area of the cut have been destroyed, cells near the cut have been damaged, and the destroyed cells release certain chemicals, the damaged cells release certain chemicals, and remember in one of the earlier lectures, I showed you that almost half of all proteins in your cells are involved in cell signaling, communication among cells. Well, the cells around that area of the cut will get those signals. They'll get those chemicals. They'll recognize those chemicals as signals and say, okay, there's been damage. And not only that, but they'll be able to tell where the damage is because they can tell where the chemicals are coming from. So what they then do is they get out of G0, go back into G1, and they start to do two things. They start moving towards the area of the cut, and they start dividing. And the whole purpose of that is to prepare themselves to repair that damage to the skin. Okay, so let's say a cell then gets that signal. It goes out of G0 back into G1 and immediately checks to make sure that everything is all set and ready to go. And when it does that, it passes then this little thing which is called the G1 checkpoint. Now the G1 checkpoint is a really critical checkpoint in the cell's life cycle. The cell will only go through it if it's committing itself to divide. The moment it makes that commitment, it has one of two fates. It's either going to divide or it's going to die. Those are the only two things that can happen to a healthy cell. So at that G1 checkpoint, it's checking to make sure that it's ready to go, that no amount of damage that occurred when it was sitting there in G1, and again, it could have been sitting there for 20, 30, 40 years, no amount of damage that it accrued in that time interval is going to keep it from being able to replicate properly. So it checks the following things. It checks whether or not the size of the cell is adequate. It's a big enough cell. It's got sufficient nutrients to divide. It's getting enough stuff from the blood and so forth. It's got the social signals. Remember I talked about the signals that are telling it to divide or not divide. That's what the social signals are. And that the DNA, it checks its DNA to make sure it's not too damaged. Because if the cell finds that its DNA is damaged and it tries to replicate, it can generate diseases like cancer. So what it does is it checks all those things. If it fails any of those checks, say for example, for some reason the DNA is quite damaged and it discovers, the cell itself discovers that its DNA is damaged, then the cell will guarantee it'll kill itself if it's healthy. It goes through a process that we call programmed cell death. It's cell suicide. Literally, it uses its mitochondria and it just releases all these chemicals that chop up its proteins and destroy it. So literally it will commit suicide if it's not ready to go. So that's why the G1 checkpoint is important. The cell's not going to check unless it's really sure that it needs to actually continue. Now, if it passes, in other words, it passes all these tests, then it commits itself into the next phase, which is what's called the DNA synthesis phase, which we shorten to S. In the synthesis phase, it's doing exactly what we would expect that word means. It's taking the DNA and copying it. Okay, now, here is something that we have to kind of think through. If we go back to this picture up here, we go to these chromosomes, and we look at a chromosome like this, this is the idea that most people come to this class with of what a chromosome is. This chromosome is not the type of chromosome that we have in G1. In G1, there's only one chromatid. There's not two. And the reason is because the cell will only make a second chromatid if it's prepared to divide, if it commits itself to divide. So throughout G1 and G0, there's only one of these chromatids. Let me show you that in a different way. If we go to this picture here, you'll see that. Now this is the chromosome. You see it's long, skinny thing. It's not condensed because, again, this chromosome is only condensed when it's preparing to divide. So this chromosome is what it looks like when it's in this configuration here, like that. Okay? So there it is. The centromere is still there. And you've got this now single strand of DNA, and that's G1 and G0. But now the cell has checked the G1 checkpoint, goes into the synthesis phase, and in the synthesis phase, it takes this DNA and copies it completely. And when it copies it, you'll see here in this picture, there's two parallel strands. And there's that centromere that's connecting the two together. Okay, so it makes a copy of every single gene on its sequence. All right, so that's all it's doing. Now we're right here in this point. Don't go anywhere else. Don't go further. Right here, we are in S. This is what S is doing, is making those copies all the way through. Now, when it starts the S phase, when it starts that synthesis phase, it begins a timer. It turns a timer on. The timer starts ticking. And it has a certain length of time to get through all of that DNA and copy it, as well as prepare for division in other ways. For example, it has to then also copy all of its organelles, it has to make copies of a bunch of other stuff, and prepare itself for the actual rigors of division. That's a very, very rigorous, dangerous sort of time for it. So it prepares itself, and that second preparation phase is called the G2. The whole time it's going through S and G2, that timer is ticking away. Then the alarm goes off on that timer. And that's it. That's the time that the cell has allotted itself to get all that stuff done. At the time that it gets to that trigger point, we go to what's called the G2 checkpoint. In the G2 checkpoint, the cell then stops everything and double checks to make sure the chromosomes are being completely successfully replicated, and they've done so in such a way that there hasn't been a lot of DNA damage. One of the things you're going to learn in your upper division cell biology class is that while the DNA is being replicated, it's also being checked. And there are sometimes the check proteins, the proteins that check to make sure the replication is done correctly, are broken. And when that happens, then you can introduce an enormous number of mistakes into the DNA, and it'll check it here and make sure that those mistakes aren't there. It also checks to make sure that the mitogen protein factor is ready to go and everything's ready to actually begin the actual division process. So it's checking all those things. If it fails any of those checks, guess what? It kills itself. It goes through that same programmed cell suicide that we talked about earlier. Okay, so when I said the cell's either going to divide or it's going to kill itself, this is what I meant. It checks to make sure that everything's going the way it's supposed to go. Now, again, this is healthy cells. So if it passes that G2 checkpoint, everything's done, the DNA's replicated, everything's ready to go, then it will enter mitosis. Now, In lab, we're going to study mitosis. I'm not going to study it in lecture, but in lab, we'll study mitosis. You've seen it before. This right here is the beginning of the first phase of mitosis called prophase. Then prometaphase, metaphase, anaphase, telophase, all is in this little tiny blue arrow right there. Okay? Now, as you're going through mitosis, the cells will condense their chromosomes, and they condense the chromosomes into the structures that we generally think of as chromosomes. These things look like this. They look like the X's. Right? So the cells then do that. So here we have the G1 phase, there's G2 and S, they copy. Then during mitosis, they condense the chromosomes. The chromosomes then split, the two chromatids split, and we make daughter cells here. And that's what mitosis and cytokinesis are going to do. And then we got two baby new cells right here. But before we go through all of that, or during the time we go through all that, there's stuff that the cell's doing, is checking on itself. So it starts at prophase, condenses the chromosomes. Prometaphase, the nuclear membrane opens up, and the chromosomes spill out and the nuclear membrane disappears. In metaphase, all of the centromeres line up in the middle of the cell, and the cell stops. And it checks. It has another third checkpoint here. And that metaphase checkpoint is making sure that all the chromosomes are attached to the what's called the spindle apparatus, which is controlling how the chromosomes move, and making sure that they're all right in the center where they're supposed to be. And if they pass that test, then they continue on into anaphase, where they split the two sister chromatids, as we're going to study in lab. Guess what happens if they don't pass? If there's one chromosome sitting off on the side, and it's not attached to the spindle properly, the cell kills itself using that same programmed cell suicide. So that is the whole point. That goes through this whole cycle, but again, it doesn't go through it and go through it and go through it. It goes through the cycle, but then again, it always defaults into G0, and it won't go through the cycle again until it's told to do so, unless it becomes pathological. And there are a number of different diseases that can cause the cells to just simply go through the cycle over and over and over again. One of them is being cancer. In cancer, one of the things that happens, one of the many things that happens in order to have this occur, is these checkpoints all have to be broken. The machinery that double checks everything and causes apoptosis, causes that programmed cell death, has to be broken, among other things. So that's the basis of our cell replication cycle. The thing I want you to keep in mind is when we get through G1, we always go to G0 unless we're told not to, and we have these three checkpoints. If we fail at any of those three checkpoints, that cell will kill itself. All right, so going back to development, that process of cell proliferation, which includes mitosis, cytokinesis, and all the cell cycle, is how we get all the different cells that we need, and all of them having all complete sets of genes. But the question then is this: are we just one giant lump of 30 trillion cells of the same type? And the answer is no, we're quite a bit different. In fact, if you look inside a living human being or any other mammal, you're going to find about 200 different cell types. Neurons, hepatocytes, renal cuboidal cells, all sorts of different types of cells that are making up all the different tissues out of which our body is made. But you can take a neuron, which is this long, skinny thing with a bulb at one end with a bunch of things sticking off, and compare that to, for example, a kidney cell in what's called the proximal convoluted tubule, which is just a box. And you look at the genes in those cells, and they're identical. They're absolutely identical. So the question is, how is it that we have cells with exactly the same genes, they're clones, but the cells look wildly different so that we can get 200 different kinds? That process is what we call differentiation. And here's an example. We start out here with something called a mesenchymal stem cell. Now, a stem cell is something we're going to talk more about later in the course, but here's what I can tell you right now. If your understanding of the phrase stem cell comes entirely from the internet and the media and newspapers and things like this, almost certainly you don't really know what a stem cell is, because very, very few people out there actually talk about it correctly. But here's what a stem cell is. By definition, a stem cell has the ability to divide into two different types of cells. We call it asymmetrical division. One of them stays a stem cell, and the other one differentiates. That's the key to a stem cell. So this mesenchymal stem cell has that property. It can divide, and one of its daughters will stay a stem cell, and the other one will start to differentiate, start to change in its structure and function, and start to commit itself to some particular type of function. Okay, now This is a special type of stem cell, mesenchymal, meaning it can only give rise to the cells that we refer to generally as mesenchyme. For example, these cells can go through this pathway and become bone cells, osteocytes. They can also go through a different pathway and become chondrocytes, which are the cells that make up the cartilage and produce the cartilage. They can also go through myogenesis and become myotubules, which are skeletal muscle cells. They can go through another sequence of divisions and differentiation and become marrow cells, bone marrow cells. They can also go through a series and become tendons and ligaments. They can become adipocytes for adipose. They can become other types of connective tissues and so on. But they can't specifically become like neurons, for example. They can't become hepatocytes. They can't become cells that line your mouth or your nose. They're stuck into becoming one of these. So therefore, they're not totipotent like the cells we saw in the morula, which are stem cell-like, and they can give rise to everything. Okay, so these are what we call pluripotent. They can go to a bunch of different things, but not everything. That process, again, is called differentiation. And again, that mechanism by which that occurs is not based on the fact that they have different genes. All of these cell types have the exact same genes in them. The difference between an osteocyte and a myotubule, or an osteocyte and an adipocyte, is which genes are turned on at different times during development. That determines its differentiation pathway. We're going to talk about that in, again, great detail later on in the course. But that's the second process. That's the process by which you get all these different cell types. But there's a third thing that we have to solve in order to build a human body out of one cell zygote. And that is, we've got to take all these 200 different types and organize them into things like this, a hand or a heart or a brain. Structures that have particular functions based on their actual anatomical positions. And that process is called morphogenesis. There's the term. Now, this root, morph, may not mean what you think it means. Back, I think in the 1970s or 80s, there was a movie with Arnold Schwarzenegger that was called Terminator. It was the first Terminator movie. And in that movie, they did a special effect where they had individuals that would look like one person and then go through this kind of really cool transformation. Actually, I think it was the second one. Go through this really cool transformation where they look kind of like liquid mercury and then they become something else. And they called that term something, I don't remember what it was, but it was shortened into morph. They morphed from one thing to another. And so nowadays we use the word morph to mean change. But the word morph itself does not mean change. The word morph means form or shape. So morphogenesis is the old way of using that word. Morph is literally meaning form, and this is the production, genesis, to generate the form. And these kinds of processes are also critical. You have to not only have all these 200 different cell types, but you have to have them organized in the right way. And some of the ways that this occurs is kind of interesting. It's a different sort of process than what you might think. Let's go back up here and let's take a look at these developing embryos. And you see right there, there's that front limb bud. And the question is, here is where the fingers are going to develop. And the question then is, why or how do those fingers develop? Here's not what happens. You've got this paddle, as you can see in that picture, and it does not occur that you get 5 different little knobs start to grow out like that and make a hand. That's not what happens. What happens actually is this. You have this bud that forms as sort of a paddle, and then 4 lines of cells, which you can see they're highlighted in such a way that they turn black here. They start turning on that cell suicide program I mentioned earlier. And they start committing suicide, and they die back, and they die back, and they die back, until eventually the fingers will then separate. There you can really see these cells right here are dying at a very, very high rate, until eventually the fingers separate. And that's true in all vertebrates. All vertebrates will end up eventually with 5 fingers. We'll see that later. That process that I've been calling programmed cell death or programmed cell suicide has another term. It's called apoptosis. Apo, and then PTOSIS is the root. Okay, now, notice, you look at that and say, no, it should be pronounced apoptosis. Many people do. A lot of professional scientists call it apoptosis. There's an argument about whether or not it should be called apoptosis or apoptosis, and I don't care which you call it. You can call it either one you want. It's fine, no problem. I am going to defend myself for calling it apoptosis, however, because of the way the word is formed. Apo means above. Ptosis is a word, Ptosis. Now, let me ask you, what is the name of the giant extinct flying reptile? Okay, extinct flying reptile from the dinosaur period, right? Well, that's a pterodactyl. The first letter is not T. It's pronounced pterodactyl, but the first letter is P. The P is silent. Okay, now, Ptosis, same thing. The P is silent, so it's ptosis, not ptosis. Right? Now, I'm not saying you can't pronounce it apoptosis, so people don't email me bad things. I'm just saying I'm defending why I call it apoptosis. Right? That's all it is. That's all that's going on. I also recognize there are reasons why it is you can call it apoptosis if you want. For example, that root, PTER in pterodactyl, pterodactyl means wing hand. PTER means wing, dactyl means hand. So if you look at, for example, the heavier than air ship with a rotary thing that spins around and lifts it off the ground, you know that as a helicopter, right? Well, break the word down. Helico means to spin, and it's that same root of wing, PTER, which is ter, which means spinning wing. Helicopter literally means spinning wing, but it really, in some sense, should be pronounced helicoter. Okay, now we can argue about that, but I'm not going to say you should pronounce it helicoter any more than you should pronounce this apoptosis. If I'm not saying you have to do that, but I'm going to continue to say I'm going to continue to say apoptosis. But the point is, this is it. This is this programmed cell death, and it's a critical aspect of a lot of biology, not just morphogenesis, but also cancer biology. But anyway, those are the three processes. That's how we get a bunch of new cells. That's how we differentiate them and change them into different forms, even though they have the same genes, and then starting to organize them into structures that are morphed. They begin to form these structures, these forms.